आइए अब हम बात करते हैं रीजेंट्स एंड सप्लाइज रिक्वायर्ड फॉर फॉर्मेल्डिहाइड एगारोज जेल इलेक्ट्रोफोरेसिस। अगेन जो ज़रूरी बात है कि जो भी हम सॉल्यूशंस बफर्स बनाएंगे दैट शुड बी प्रिपेयर्ड इन DEPC ट्रीटेड वाटर। हमें एथिडियम ब्रोमाइड की ज़रूरत पेश आएगी 200 माइक्रोग्राम पर मिलीलीटर कंसनट्रेशन, अगेन प्रिपेयर्ड इन DEPC treated water. Formaldehyde is supplied as a 37% solution, weight by volume, and it may contain some stabilizers like methanol, क्योंकि formaldehyde जब expose होता है air के साथ it is oxidized to make the formic acid। और लैब में अगर हमारे पास फॉर्मेल्डिहाइड मौजूद है, इसकी pH और इसका कलर जो है दैट कैन इंडिकेट वेदर इट नीड्स टू बी deionized before it can be used or not. Its pH, if it is acidic, that is less than 4, or if its color is yellow, then before using the formaldehyde it should be deionized using mixed bed resin. Formamide can be purchased from commercial source and it can be stored under the nitrogen at -20 degrees centigrade in the form of small aliquots. So the first preparation is 10x formaldehyde gel loading buffer. Formaldehyde gel loading buffer will be used to mix up with the RNA molecules, RNA samples to prepare the samples for loading into the gel. इसके लिए हमें ग्लिसरॉल की ज़रूरत है, EDTA चाहिए, ब्रोमोफिनोल ब्लू चाहिए और ज़ाइलिन साइनॉल FF की ज़रूरत पेश आती है। अब इसमें अगर ईच कंपोनेंट का हम देखें कि क्या फंक्शन है, ग्लिसरॉल विस्कोसिटी इंक्रीज़ करने के लिए है, ब्रोमोफिनोल ब्लू इज़ रिक्वायर्ड टू गिव द कलर टू द सैंपल सो दैट वी कैन ट्रेस दैम। हमें इलेक्ट्रोफोरेसिस बफर की ज़रूरत पेश आती है। 10x MOPS buffer is going to be used. Again, the concentration is given, the molar concentration is given in this slide and the recipe is given in this slide. How we can prepare specific volume of 10x MOPS using 0.2 molar MOPS, 20 millimolar of sodium acetate and 10 millimolar of EDTA will be the composition of 10x MOPS. Agarose gels containing 2.2 molar formaldehyde. अब volume जो है that is decided depending upon the size of gel which will be casted. If the electrophoretic chamber is small, medium or large, accordingly हम जो volume है gel की वो adjust कर सकते हैं। इस वक्त हम जो बात करेंगे we are going to talk about 100 milliliter of the volume. To prepare 100 milliliter volume of the agarose 1.5%, we are going to take 1.5 gram of the agarose and add it in the 72 milliliter of water. Agarose cannot be dissolved in water at room temperature, so we have to microwave it or we have to heat it to boiling so that agarose gets dissolved, then cooling it down to 55 degrees centigrade. The next step would be addition of MOPS electrophoresis buffer and the deionized formaldehyde. Addition of the formaldehyde should be done in the fume hood and then we cast this gel in the gel casting tray using a comb. Required number of wells को सामने रखते हुए हम comb select करते हैं। 3 millimeter की comb select की जाती है। This gel will be left in the fume hood for 1 hour or 60 minutes so that it gets solidified. And once it is solidified, it can be used immediately or it can be covered with the cling film so that we can prepare the samples. The next thing required is RNA samples. RNA samples are prepared from the purified RNA, whether this is total RNA or this is the purified poly-A RNA. Again, we have to be assured that there should not be any salt concentration, ion concentration or SDS present, and the concentration or amount of RNA should not be more than 10 micrograms, because more than 10 microgram is going to make the streaks in the electrophoresis. So in a volume of 1 to 2 microliter, the 10 microgram of the RNA is taken, and then it is added up with the RNA loading buffer to proceed further. The special equipment required is a horizontal gel electrophoresis chamber. Optional is the ruler to measure the distance traveled by any particular RNA band, and then the water bath, which is preset at 55 degrees centigrade. It will be required for some specific incubations.